So, as you have noticed, I am not in my typical setting with my whiteboard behind me, and that's because it has been a dreary Monday, and we're now into the evening where it's been cold and rainy and not fun. And I felt like I really needed to build a fire because fire adds +5 to spirit. So this is where we are. I think we could all use a little extra spirit. So tonight I thought I would just do a little fireside chat with you instead of the typical lecture. And we'll kind of just talk through transcription and translation. If I'm feeling ambitious, I will try to put some words up on the screen during the video editing process, but no guarantees that they're going to be all of the words that you need to know. So make sure you're taking really good notes throughout this video, and if you have any questions about expectations, just assume if it's a word that you didn't know before that you should know it. And of course you can always email me. So, wherever you are, whatever you're doing right now, if it continues to be chilly and gross for the next couple of days as you are going through this week's topics, be sure to cuddle up by a fire if you can, and if you can't, be sure to put on a YouTube video that has a fire in the background so that you can feel good about it, or just watch this video multiple times. Whatever you need to do. Also, grab yourself a cup of tea, which is what I have done to make this whole experience more pleasant and what we all need right now. So, let's get started with talking about transcription and translation by talking about transcription. When we're talking about transcription, we're talking about that process of taking genetic information from DNA and using that as the blueprint to create RNA. And specifically, we're talking about the creation of mRNA, or that messenger RNA that we talked about last week. And this is really important for being able to go on to that next step of translation where we're going to create proteins. So this is sort of the first step. Now, it's easy to confuse the terms transcription and translation because they're close to each other, but think about it this way. What is happening in transcription is information is being transcribed from DNA into RNA. So we're transcribing something into a different language, but we're not creating anything. With the process of translation that we'll talk about in the second half of this video, you're actually creating a product. So you're translating that language into something meaningful, into the creation of proteins. So that's one way that you can think about how do I differentiate between transcription and translation. Transcription, you're just changing the language. Translation, you're making that language into something tangible. So with the process of transcription, which we'll start with first, because it's the first step in the process, which makes sense, you're going to start with DNA. Remember that DNA is going to be what encodes for and helps us to create these strands of complementary messenger RNA or mRNA. So for every DNA nucleotide that is read, we're going to add a nucleotide onto the strand of messenger RNA, and it's going to be complementary to whatever it's reading from the DNA. So as we start thinking about this process with transcription, I'd like to start by having you all just sit back and unwind a little bit, because unwinding is exactly where we're going to start. The DNA double helix is going to be unwound, and then you're going to have two strands. You're going to have your template strand, which is going to be used to create the messenger RNA, and then you're going to have a non-template strand, which I bet you can guess what that does. Nothing in this whole process of transcription. We're going to focus on the template strand. But one thing you should keep in mind about the non-template strand is that it's going to be virtually identical to the messenger RNA that is created from the template strand because it's going to be complementary. Of course, it's going to have a U where it normally would have had a T if we were talking about the non-template strand of the DNA. Now, this process of transcription is fundamentally the same between prokaryotes and eukaryotes. So we're going to kind of talk about this in general terms, but there are some slight subtle differences between the two different kinds of organisms when we're talking about these processes. And I'll do my best to kind of point out those differences as we go along. One of the big fundamental differences about this process and about the process of translation that we're going to talk more about here in a little bit is that it's happening in different places in prokaryotic cells versus eukaryotic cells. So we know with prokaryotic cells, they don't have a nucleus. So all of this material is being transcribed and translated in the cytoplasm of the bacterial or the archaeobacterial cell. So we're just getting transcription and translation that is occurring, kind of hanging out in the cytoplasm, all willy-nilly like. It's a laissez-faire, crazy kind of time inside the prokaryotic cell when we're talking about these processes. Now when we're talking about eukaryotic cells, it's a little bit different. It's a little bit more compartmentalized and regulated because most of this stuff is happening in specific areas of the cell. So this first process of transcription is going to be something that's occurring in the nucleus of the cell and isn't going to be just happening out in the cytoplasm. Now that makes it slightly slower than what we have in a prokaryotic cell, but it also makes the molecules much more stable. So that's one of the trade-offs in eukaryotic versus prokaryotic cells. Now, once we have a product that has been transcribed inside of the nucleus of a eukaryotic cell, and we've got our nice little strand of mRNA, that mRNA is going to be transported out of the nucleus and into the cytoplasm where it's going to be finding some ribosomes to be able to continue on this next step of the journey in protein synthesis. This can be in the cytoplasm itself, where we've just got ribosomes floating around, or it can be found in the endoplasmic reticulum, specifically the rough endoplasmic reticulum, where these ribosomes can be hanging out inside of these eukaryotic cells. So we're going to be able to continue on to that process of translation in one of those two spaces within the eukaryotic cell. There are some specialized types of machinery, and we're not really going to get too deep into it because this is kind of an overview part of the lecture, but later in the semester, as we start talking about biotechnologies, we'll start talking about some of the specifics of the transcriptional machinery that is used for these processes. And we won't say too much about enhanceosomes, but let's just say that they are important for being able to recruit some of that transcriptional machinery to where it needs to be so that we can get this whole thing going. These enhanceosomes are proteins that bind to the DNA and bend the double helix in one direction. Now, just like with DNA replication, there are going to be three major phases to this process of transcription, and they've even got the same names: initiation, elongation, and termination. You're going to see those three terms popping up again here in a little bit when we talk about translation. Now, the process of initiation is going to begin with DNA unwinding so that we can open up a site where we're going to begin the synthesis of messenger RNA strands. The region of the DNA double helix that is starting to unwind is going to be an area that we call the transcription bubble. And it's within this transcription bubble that we're going to see the creation of that mRNA strand, or that whole process of transcription taking place. Within that transcription bubble, as the DNA is unwound, you're going to wind up with your template strand and your non-template strand like we talked about before. Your template strand is going to be what is read for the process of putting down the nucleotides in the correct sequences to form the messenger RNA strand that is going to be complementary to that template strand. Now, on that template strand, you're going to have one region known as the promoter region, or simply just known as the promoter. And this promoter is going to be a sequence of the DNA where some of the different machinery that begins this process of transcription is going to bind. This promoter typically exists upstream of the genes that we want to encode to be able to create our messenger RNA. When we use the term upstream versus downstream in terms of DNA, we're talking about directionality. So upstream would be something that is encoded first, and downstream is something that is encoded later. So our promoter is going to be located upstream of the sequence of DNA that we want to transcribe. Now, the second step in transcription is going to be the elongation step. And as the name implies, this is going to be where we're elongating the messenger RNA strand by adding additional nucleotides to the sequence based on the genetic blueprint that is set forth by the template strand of our freshly unwound DNA. A special enzyme known as RNA polymerase is going to bind and proceed along the template strand of the DNA to be able to create that mRNA strand. Now, unlike in the process of DNA replication, the mRNA strand is not going to remain bound to the DNA template. Instead, that mRNA is going to be free. As elongation proceeds, the DNA is going to be unwound ahead of the RNA polymerase, and then behind it, we're going to see the DNA helix being rewound into its original configuration. The final step of this process is known as termination, and like the name implies, this is going to be the end step of that transcription process. There are going to be a number of termination signals that are going to be involved in instigating the beginning of the end of this process. There will be a series of repeated nucleotides that are going to stall out the RNA polymerase, cause it to leave the DNA template, and eventually free that little mRNA transcript that was created during the process. In prokaryotic cells, where we're not constrained to working inside the bounds of a nucleus, we're going to see that these polymerases can transcribe and then move straight into translation pretty quickly. In fact, those two processes can occur simultaneously within the cell, and multiple polymerases are going to be involved at a time. So they're able to really create some protein products very rapidly, and they're going to be able to reach really high protein concentrations within the cell in a very short time period. This is a property that we use to our advantage quite a bit with molecular biotechnology, so we'll be returning to that later in the semester. Now, eukaryotic mRNAs are of course different because they're going to be transcribed in the nucleus and then transported out of the nucleus to be able to go through that translation process in other parts of the cell. And to be able to make that journey, we have to modify the mRNA a little bit to keep it from getting degraded. This process of preparing the mRNA for that journey outside of the nucleus is something that is often referred to as maturation. And just like we find in human beings, the more time something has to mature, the more stable it's going to wind up being. This makes eukaryotic mRNAs much longer lasting than prokaryotic types of mRNAs. Many eukaryotic mRNAs can last for up to hours, whereas by comparison to our prokaryotic RNA friends, those things are lasting pretty briefly, sometimes fractions of a second. Much like a terrible lover, most of the mRNAs that are created in prokaryotic cells don't last more than about 5 seconds. Now, going back to our eukaryotic cells, we know that transcription and translation are separated by time and space because of where these processes are taking place within the cell. And like we've said before, those mRNAs that are coming out of the nucleus have to have special adaptations to keep them from degrading as they make that journey out of the nucleus into the other parts of the cell. Preparing these mRNAs is known as RNA processing, and it starts with coating the mRNA in stabilizing types of proteins. First, a nucleotide cap is going to be added to the 5' end of the mRNA transcript. This cap is also going to be useful later on for the initiation process of translation. To the opposite end of the mRNA transcript, in other words, the 3' end, we're going to see a series of repeated adenines. So this is going to be what we call the poly-A tail. And the poly-A tail is important for signaling the end of that process. And that poly-A tail is going to contain upwards of 200 or more adenine residues. This repetitive sequence acts as a signal, bringing in factors that help with that process of exporting the transcript out of the nucleus. You can kind of think of it as the cell's way of calling an Uber for the mRNA transcript. There's also a really important process known as RNA splicing. So within the mRNA transcript, you're going to have some areas that are known as exons, and those areas are going to encode directly for amino acids. In between all of those exons, you have these little areas of non-coding genes that are known as introns. To be able to move forward with the translation process smoothly, we've got to remove those introns and get all of our exons together. So this is a process known as RNA splicing, where with great precision, we're going to take out those introns and scoot our exons close together to be able to be more efficient in that process of translation later on. This is happening while the mRNA is still inside the nucleus, and those little introns are just going to get degraded within the nuclear space. There are a lot of different factors and proteins that are involved in this whole process of RNA splicing. Some of these things are going to be what we call spliceosomes, and you'll hear that thrown around in the literature as you move through learning more about this process. Once an mRNA has been appropriately sliced and diced, then and only then is it going to be capable of moving outside of the nucleus. Alright, so that brings us to this process of translation. This process of translation, which again is where we're translating an mRNA into a physical protein product. That process is going to be one of the most energy-consuming metabolic processes that the cell is going to undergo during its lifetime. Remember that proteins account for a huge amount of the mass within the cells, and that they're really important because they perform a lot of different functions like we talked about last week. So this next step in being able to create those proteins is super important. Our messenger RNA is going to encode for those different proteins, but to be able to create those different proteins, that mRNA is going to have to be decoded and translated into the amino acid sequences that will eventually form our polypeptides to create these different proteins. This process of translation is going to require the input of mRNA template strands, as well as involvement of the ribosome, a wide variety of enzymes, and our old friends, the tRNAs, also known as the transfer RNAs. Now, ribosomes are going to contain polypeptides as well as ribosomal RNAs, or our little friends, the rRNAs. In eukaryotes, those rRNAs are going to be found within the nucleolus of the nucleus. And these little ribosomes are going to be comprised of two major parts, a smaller ribosomal subunit and a larger ribosomal subunit. In the smaller ribosomal subunit, that's where we're going to see the binding of the mRNA template. The larger ribosomal subunit is where we're going to see the binding of our tRNAs, which are going to bring amino acids into the growing polypeptide chain as we form these different proteins. mRNAs are going to be translated simultaneously by multiple ribosomes to be able to create these proteins. The types of tRNAs that are involved in this process are going to be variable depending on the species of organism that we're talking about. You can have between about 40 and 60 tRNAs involved in this process. It's these tRNAs that do the actual translation part, taking the language of the mRNA and translating it into the language of the proteins. It does this by pulling in amino acids and being able to place them in the appropriate place at the appropriate time based on the template that it's getting from the mRNA strands. In order to do this, tRNAs must be charged with specific amino acids. When we use the term charged, this is just to refer to a tRNA molecule that is bound to a specific amino acid. Now, you remember our friends, the amino acid, how we had 20 of those? You can kind of think of those 20 amino acids as 20 letters in a protein alphabet. Each of those letters is going to be defined by a 3-nucleotide sequence that is known as the triplet codon. Each of these sequences of 3 nucleotides is going to encode for one specific amino acid. This relationship between that nucleotide sequence and the corresponding amino acid is going to be referred to as the genetic code. And the cool thing about this genetic code is that it's pretty much universal across different organisms, both eukaryotic and prokaryotic in nature. There are a couple of exceptions to this that I'll talk about here in a second, but they're pretty rare. This genetic code was revealed in 1968, and it was discovered as we completed the genome for the bacterium known as E. coli. Following this discovery of the genetic code in this little type of bacterium, we started to see the same code popping up in other organisms, more complex organisms. So this was really strong evidence for the argument that all life evolved from a single organism if you went far enough back on the evolutionary tree. Most of the exceptions to this universality that we see in the genetic code lies within a mitochondrial genome of different organisms. Mitochondrial genomes are going to be pretty different from nuclear genomes. We're going to see some subtle variations in things like Drosophila melanogaster, which is a fruit fly and one of the most studied model organisms in the field of genetics. Also in a really cool bacterium known as Mycoplasma capricolium, as well as a variety of different protozoans. When we look at the genetic code, remember that we are working with 4 different types of base pairs, and these are going to be able to exist in combinations of 3 sequences at a time. So you're talking about a possibility of 4 times 4 times 4, which gives us a set of 64 possible combinations for our genetic codes. Now remember, we only have 20 amino acids, so you're going to see some of these different codons coding for the same amino acids. And there's a really great chart in your OpenStax textbook that shows what some of these different combinations create. Now, as you start looking at these different triplets, there are 4 that should really draw your attention. 3 of these triplets create what are known as stop codons, which are going to be the signal for being able to initiate termination of the translation process. This is going to stop protein synthesis and release all of the translational machinery that was involved in keeping the process going while we were building the proteins. You'll also notice that there is a specialized form of methionine that gets created when you use the triplet AUG. This is what is also known as the start codon. And the start codon initiates that process of translation, and it also sets the reading frame for all of the triplets that are going to come after it. Right now, we're not really going to talk about what can go wrong in this process, but you can imagine that if the reading frame gets off by a single nucleotide, it can throw off the rest of the translational process by accidentally creating the wrong proteins using the wrong triplets. As in the first process that we talked about earlier today, there are going to be 3 major steps in the process of translation. And I bet you can guess what they are. I'll wait. Yeah, that's right. Initiation, elongation, and termination. Initiation is going to start by forming a complex. This complex is going to be formed by the small ribosomal subunit, an mRNA template strand, a tRNA initiator, and a variety of other initiation factors. This initiator complex is going to interact with the start codon and get set for that reading frame of being able to continue with the process. This is going to link to that special version of methionome that's going to be removed once the translational process is complete. So our tRNA anticodon is going to recognize the codon on the mRNA. The large ribosomal subunit is going to join on to the small ribosomal subunit, and a second tRNA is going to be recruited as part of this initiation process. Now we can move on to that process of elongation. This is going to involve that large ribosomal subunit, and we need to be thinking about what's happening within that subunit in three specific areas, known as the A site, the P site, and the E site. The A site is where we're going to bind our incoming charged tRNA molecules. The P site is where we're going to find charged tRNA molecules that have amino acids that have bonded to the growing polypeptide chain but haven't quite disassociated from the tRNAs just yet. The E site is going to be where we're going to see the release of those disassociated tRNAs so that they can be recharged with more amino acids. During the translational process, the ribosome is going to shift one codon at a time. This catalyzes the process that's happening at each of the three sites, the A site, the P site, and the E site, at the same time. So, at each step, we're going to have a tRNA that is just now entering the complex. We're going to have an amino acid that is being added to the chain of the growing polypeptide. And we're going to have an uncharged tRNA that is departing. There's an incredible amount of energy that is expended for this entire process. But instead of using ATP like we see in other parts of the cell for various cellular processes, this is going to involve GTP instead. As that mRNA strand moves relative to the ribosome, we're going to see the creation of this polypeptide chain and eventually the creation of our protein. This translational apparatus that we find inside of E. coli can add an amino acid every 0.05 seconds. And that translates into a 200 amino acid polypeptide sequence that is being created every 10 seconds. Finally, the last step in this process, our old friend termination, is going to involve our new friends, the stop codons. And again, there are three of these: UAA, UAG, or UGA. Once the translational machinery encounters one of these stop codons, it's going to cause the release of that growing polypeptide chain and also going to dissociate the large and small ribosomal subunits from one another. This is going to leave our old friend, the mRNA template strand, just sort of hanging out. And it's eventually just going to get degraded. It gets broken down into its component nucleotides that can then be recycled for other cellular processes later on. And that's pretty much our whole process. Transcription to translation. We started with a DNA blueprint and we're able to transcribe that language of DNA into the language of RNA to be able to create our messenger RNA, which we then had to put a cap on and throw a tail on to keep it from getting degraded as it made its way outside of the nucleus to be able to go into that process of translation, unless we were in a prokaryotic cell, in which point we didn't have to worry about that because it wasn't in the nucleus to start with. And then that messenger RNA is going to go out, it's going to find a nice ribosome to call home, it's going to hook up with a tRNA and be able to create these cool little polypeptide chains that eventually go on to be our proteins that are so important for all of these different things that are going to be happening on a cellular level. So just a couple of little housekeeping things for you guys for this week. One, your highlights as usual are going to be due to me by 5 o'clock on Friday. Be sure and get them to me on time. We are going to have an additional reading that I think you will thoroughly enjoy that came out in 2008, and it is looking at the evolution of genetic coding. So thoroughly enjoy that paper. And from that paper, I want you to take 3 things away and add those 3 takeaways into your highlights for this week. So exam number 1 is going to be coming up before you know it, and that is going to be administered through D2L. I will have it open for 24 hours. More details will be coming about that next week. After exam number 1, we're going to have some guest lectures come and join us for a couple of different topics throughout the weeks that we're talking about molecular biotechnologies. So be on the lookout for that. We are going to work on the time schedules of our lecturers since this is an asynchronous class. If you can be there live, that would be awesome. so that they're not just talking to me in a recorded session for Zoom. I will let you know about the scheduling for that as we are able to lock people in. We do have some people who are coming from very different time zones. For example, one of our guest speakers is in Finland, so we're going to have to kind of play around their schedules with that to be able to be fair to them and conscientious of their time. So, if it works in your schedule to be there for the live session, I want you to be there. If you cannot be there, I expect you to watch the recording afterwards. And whether you're there live or you catch the whole thing on the replay, I'm going to expect you to take some of your major takeaways from that conversation and incorporate them into your highlights for that week's assignment. I'm also going to be giving you some information about our guest speakers and their expertise and the kinds of things that they're going to be talking about. And I'm going to ask you to submit to me at least one question ahead of time. And that's going to be true for everybody, whether or not you can be there. If you can be there in person, you can ask your own question. If you can't be there in person, I will ask your question on your behalf. Also, great job on the discussion boards lately. We're starting to see a lot more productive conversation, but I encourage you to keep going with it. Keep talking to each other, keep engaging in conversations. Again, I am purposefully not stepping into these discussions, but I am very creepily lurking and reading everything that you are writing to one another. So please do engage with your fellow classmates in those wonderful conversations about some of these different topics that we've been covering in lecture. I also wanted to remind you that in Chapter 5 of your OpenStax textbook this week, you may have seen a QR code and or the link to a pretty cool little interactive video that can help you to look at some of these different transcriptional and translational processes and get a feel for what's actually happening. So be sure to check that out whenever you get a chance. And finally, I'm going to be sending you a lot of links to supplementary videos this week because there's some pretty cool stuff out there showing these two processes and really breaking them down and even getting into way more detail than we talked about in lecture. So please make sure that you watch at least a couple of those supplementary videos. You're not required to put anything from those supplementary videos in your highlights for the week. However, if you found something to be particularly cool, feel free to share it in your highlights and brighten my day with your perspective about those supplementary videos. So I think that is everything, and the fire is starting to die down. My tea is starting to get cold, so I guess I better finish it. But I hope you enjoyed this slightly different format. I hope you found it comforting. I hope you got Spirit +5 from the fire. I hope you stay warm. I hope you are able to continue this conversation about transcription and translation and thinking about the implications of these really important processes as we move into the next topic for next week, where we're going to look at gene regulation within both prokaryotes and eukaryotes. So have a wonderful week, have a fun weekend, drink some tea, enjoy a fire, and we'll see you next week. Bye!